I do apologize for the delay. It was a technical problem with my camera. Thanks God it's fixed. So it's always thank you for watching. I will promise every Thursday it will be another video. Thank you for support. Today, the topic is bacterial growth and development, part 1. Let's begin. Before I start the explanation, I want to say that there are a keyword signs in blue to help in understanding the main ideas in every slide. I will explain the general ideas in the slide and ask you to pay attention carefully to the written sentences to facilitate the understanding process smoothly in every slide. It is also known that the reproduction process of bacteria has requirements, as it needs an appropriate environment and needs in order to achieve the process of growth and development of bacteria. And also, I will talk about the method of bacterial reproduction, which is distinctive and different from other living organisms. The process of evolution means an increase in size and the number of cells. Focus on these two words because they are very important. An example to illustrate, a person becomes an embryo, after a child, then a young person, and then he becomes an old man. These are the stages of a human growth, and the bacteria exactly as they grow. But there are some needs that, if available, cause bacteria to grow and multiply. For example, some types of bacteria need oxygen in order to breathe, and there are some another need carbon dioxide instead of oxygen. I'll talk about it later. There are types of bacteria that need a liquid medium, and others a solid environment. In the liquid, bacteria can only be seen by a microscope. In the solid, they form colonies and look turbid. In this slide, I will talk about the bacterial reproduction process, which is an increase in the number of bacterial cells instead of size due to division, which occurs through a process called binary fission. These drawings in the slide above describe exactly how the process of binary fission, which doesn't reproduce sexually, occurs. In figure 1, the ball in the middle is the genetic material of the bacteria, and as I mentioned previously, the shapes and forms of the genetic material are different, which are linear, circular, and double strand. At this stage, the bacteria increase the metabolic enzymes and their proteins in order to increase their size. In figure 2, the genetic material becomes double and extended along the length of the cell. And the reason for this expansion is the mesosome, because it helps to separate the chromosome and push it to the end of the cell. And also, the genetic material of the bacteria is not surrounded by a special membrane. In figure 3, specifically, the cell carol, it expresses that the genetic material begins to divide vertically through the cell wall and the membrane. In picture number 4, we have approximately two separate identical cells, and each one has its own genetic material, and these two cells are formed by pinching process. In image 5 of the result, we have two completely separate identical cells. An important note, the process of reproduction varies from one cell to another. In other words, a difference in the generation time, which is the time that a cell or a group of cells need to divide. And from here, we conclude that the generation time is equal to time taken on number of generations. I hope that you have benefited from the information, and whoever has any comment, please write it at the bottom of the video on the third topic of microbiology, part 1, and I will answer all the questions. Thank you for following up. I will see you next week with a new topic. Have a good day.